So next up, we have Dr. Moica Tajnik-Zbajdzero, who leads the Virus and Nanoparticle Innovation Group at Sartorius BS Separations. Her work focuses on downstream processing of viral vectors and vaccines, with recent projects emphasizing LNP process development. She will present enhanced purification methods for manufacturing of complex lipid nanoparticle formulations with multiple payloads.
Okay, good afternoon from my side. Thank you for introduction and also describing my title. We will now move to the purification part of the LNP manufacturing. So I will show you a lot of examples we did so far in this building, and also how we combine this with the analytics approaches that were just presented by Tristan. So we will start with introducing the chromatography for LNPs, since this is a new field in the LNP manufacturing. Then I will show you examples of workflow, so how to do this in practice, and examples, and also the more detailed, while doing the process development, how you monitor different attributes that correspond to the efficient separation and purification, and how all this is combined with the switcher analytics that was introduced. So challenges we are facing, also this was introduced. So first we have the particle that is not very nice to purify. It's unstable. It's very sensitive to the shear stress. It's heterogeneous, so a lot of different LNPs for different drug products. The samples are diverse, and also there is very complex morphology, so what actually is on the surface, what the column will see at the end. So when we start the process development, here one of our challenge issue was lack of the analytical methods. So this is why first we developed the LNP switcher, because there are very limited analytics that can be used for the process development present for LNP that is going beyond the size and the activity, let's say. Then in the manufacturing challenges, so this is the most, the part of my talk, is shear stress. So this is, as I mentioned, these molecules, these particles are very unstable, so we need to always watch out not to add too much stress to the particles, and this can be a problem also in the manufacturing, especially for the scale up. We need to gain, at the end, uniform and safe particles, and also we need to choose solution and buffer that will retain the stability. So currently, the process that is used by, that was also used during the pandemics and so on, is the TFF purification. So for the downstream scientists, this is very simple purification workflow. So you have the synthesis of the LNPs by the microfluidic mixing, just dilute tangential flow filtration, and sterile filtration. So this is a relatively simple process, but simple, is not always good. So with this process, we remove mainly the ethanol, and we buffer exchange the sample to the real buffer that is then used in the drug product, concentrate, But what is the real problem with this is first scaling up, and second the shear force that is produced during the TFF. So you know the one that is using this in the lab, that you always need to, let's say, keep the constant shear rates and so on. So telling you that this is really important, this actually damage your particle at the end. And this came to our idea that actually chromatography would be the right solution. And when we heard the introduction by Roque at the very beginning, you could hear that with the large channels and the convective flow, the shear force on the column is very low. And this is the whole idea to add the purification of LNP to the monolith. So this is the, let's say, simplified version of the LNP-based pharmaceutical purification. So we have, this part was described with on the Roque talk, so pDNA and mRNA producing and purification. And then we are actually on the last part of this scheme, LNP producing by encapsulation. So encapsulating the mRNA that we heard, also the quality there is very important. And then the purification of the LNP. So let's say we are on the last stage and we need to preserve all the work that was done here and several other analytic parts and production and especially design of the payloads and all this. And we need to be really careful not to ruin this at the very end of the process. So we need to have really good LNP purification process to add to this scheme. Here in-house, we do this with control with the PATfix system. So we have platform for each of these, let's say, sides of the process. So pDNA, mRNA, and also the LNP switcher that is helping us to maintain quality throughout the whole manufacturing process. So to focus on the LNP part, so the last part, we have here very simple schematic presentation. So we have payload, lipids, encapsulate this with various techniques. And then we got LNPs that have the one that are used to some more complex bioprocess samples, very little impurities, so ethanol, non-encapsulated payloads, lipids, and we then purify this. So either with the TFF, and here we are introducing the innovative approach with the monoliths. So I already mentioned the shear stress is the main, our main, let's say, goal to lower during this purification, because it's the most important. We get at the end the active particles so that the mRNA inside will be able to express in the cells. So this is, but at the same time, we want to keep all the characteristic also on the TFF. So we want to concentrate LNPs, also remove the free ethanol and the free RNA. So we need to maintain actually the two different things during the same purification. And we do this with the hydrophobic interaction chromatography. This here is a schematic representation. So the starting point is the same, so encapsulation. Then we stabilize particles as normally, so to increase the pH, to lower the ethanol content. We obtain crude LNPs, and here the chromatography part starts. So we have then what comes out from the encapsulator, and that is neutralized. We just load straight to the columns. So you'll see here in the room many different columns. So these are actually the columns I'm talking about. And on these columns, we remove the free RNA and ethanol, and then obtain the pure and concentrated LNPs that can be, that is actually the final step of the manufacturing. We'll show how this purification workflow is done in the laboratory, and some examples of different LNPs we are purifying. So this is the real protocol, but just to show, so this is example 1 ml monolithic column OH 6 micron channel. So the channel size here is important. So the larger channels will will give better performance also regarding the shear stress and the end activity. So we recommend to use 6-micron channel. Then you use normal buffer for the neutralization. So the one you are currently using, for example, 3-SPR7.4, 10 times dilution. Then here it's important, so for loading on the OH column, you need to put some salt in the sample. So we use the different cosmotropic salts and different molarity. So this is actually one thing you need to define during the process development. You can add additives, so the chromatography performance will not be affected much also with the different additives you want to add in the final buffer. You elute in final elution buffer, so this can be your already developed final buffer for the drug product. And yeah, you add on the column, wash, elute in the step gradient to get the higher concentration. And you can strip column and reuse it also for the next purification. So for the process development, we are using detectors. So we use normal chromatography systems with the UV detector. So UV detector, and we add also our Sartorius PATfix semi-prep mouse that can be coupled on the chromatography equipment. So here is example of one chromatogram. So the one that already used chromatography can, I think, agree this is really perfect purification. So you add sample, you wash, then you elute, you get the high peak, and then here you have cleaning in place, so no peak or very small. That is actually also indicating that the capacity on the column is purely due to LNPs, so it's really high. During the loading, so here, you remove the ethanol and the formulation buffer from the encapsulation, and also the free RNA. So in case, for example, you want to also develop some continuous process or so, you can also reuse this RNA if you have higher, for example, percentage of non-encapsulated RNA in that particular process. During the purification, also some separation is achieved. So you can also study this based on your product. So I will tell a little bit more about this later. And then you obtain this elution fraction that is high functional and uniform. I will now show you with several examples. So the similar chromatogram that I'm not showing, but for example, elution here is for the luciferase mRNA 2 kb payload. We compare here the TFF compared to the OH elution. And we see here, for example, if I start on the right side with the luciferase expression, so yellow is showing OH elution, so we have a lot higher activity on the cell-based assay of the luciferase expression compared to the TFF in this case. And also when we measure this on the NTA, we can see more uniform particles after the OH elution compared to the TFF. And if we combine those two data together, we can maybe find a reason for that. So why the activity is here higher. So you can see we have also more uniform particles, means that probably due to the lower or no shear stress during the chromatography, particles were more uniform at the end. They were less damaged, and these were showing on the activity. So here we set, for example, the same amount of RNA was transfected with LNPs either with OH elution or TFF. So if we transfect the same amount of the RNA, we have higher activity on the OH because the LNP particles were more uniform after the chromatography. So the similar results for the luciferase self-amplifying RNA. So this is a lot larger RNA, 11 kb. So here we got high activity for both. So in gray is OH, and black is the TFF. So we either use the flat sheets hydrosart 300 kilodalton membrane, or the hollow fiber microflow. And we see that we got also, in this case, higher activity after the OH purification, and the particles were quite comparable for the both techniques. So either with the OH or the TFF control. When we use the dual loading, so these samples are also quite challenging for all aspects, starting from encapsulation. We see here, in this case, we encapsulate at the same time pDNA and luciferase mRNA into the LNP. We see comparable particle size distribution. So in both cases, we got, let's say, very nice LNPs, but higher activity in the OH elution compared to the hollow fiber TFF filters here. So these were the three cases. We were showing activity, comparison, and the size. And now I will move to the monitoring the process during the process development, what attributes you can follow during the process development and also later for the manufacturing. So we will mention ethanol removal, concentrating of LNPs, how much you can load on the column. So this is important, like what column size to use for the start and for the later stages, how you can scale up the process, if you get some separation also during the chromatography, and what happened with the lipids during the OH chromatography. So we were actually confirming and checking what happened with the ethanol, since this is one of the impurity that should be removed during the purification, and it's a very high concentration after the encapsulation. So you can reach like 25% or so for the at the end of the encapsulation. So this needs to be lower to the minimum or to very, very low levels for the drug product. So here we use the gas chromatography to actually check the ethanol concentration in the load samples, flow through. So this is what doesn't bind to the column, so what just flow through the column, and then the OH elution for the three different samples. And when we calculate this on the volume, because we also concentrate a lot during the chromatography, we see like nearly complete removal of the ethanol in the elution fraction on the OH column. And yeah, so in some cases below limit of detection or very, very little amount present. And this is showing also here on the graph. So this is the elution fraction, complete removal of the ethanol. The concentrating is one of the main also question, because chromatography is the last step. So it's not supposed to follow by another TFF just for the concentration. So we need to reach certain amount of the certain concentration after the chromatography to have a really clinical valuable process. So we got here, if we really if we load like a lot of the material and we do step gradients, so this is very fast elution straight to the elution buffer, we get up to 2 milligram per milliliter of encapsulated RNA in the final sample, in the final elution. And this can then be monitored, and you can also dilute it to reach your final buffer composition for the drug product. This is connected to the capacity. So you can reach certain concentration if higher concentration more near the capacity of the column you are. So we measure here for the SM102 formulation that we have the capacity 3.7 milligram of the RNA per milliliter of column. So you can load up to this amount per milliliter of column. So you can multiply this with the size of column to help you decide what column size you can use based on the amount of RNA you want to purify it in the LNPs. So if we also add the lipids, we got up to 40 milligram of the LNP mass per milliliter of column. How you obtain this value? So here, actually, the chromatogram is cutted because it should go like until here. So here we loaded like more than 1 liter to 1 ml column, and after a certain amount, your UV signals start going up because the LNP cannot bind anymore to the column, and you will see the signal that we call the breakthrough, and this is when you reach actually the capacity of the column. What about recoveries? So here in this case, we were comparing TFF process again with chromatography elution. And here on the TFF, we got quite bad recovery, 72%. But on the OH chromatography, it was 90 to 95%. And this is actually normal recovery we are getting during the purification. The scalability. So this is the calculation, like how much you can load on what size of the column. So if you have very little sample, precious material, as usually, you go to the very small column. So you can also get 0.1 ml column, where you can load like micrograms of the encapsulated RNA. And then you multiply this with in the process. So you choose different columns. And this is the Simultus line that is fully scalable up to 40 liter. And on 40 liter, you can probably purify it around 150 grams of encapsulated RNA. So this is a real scale manufacturing process. So we are also, like when we talk about chromatography, it's not only about concentrating and removal, but it's also about the separation. So we here see, for example, in the process development, we were fractionate peak of the LNPs to several subpopulations, and we analyze them for the luciferase expression and sizing. And we could, for example, remove, we could see that this first and the last fraction that we're showing here and here were less active than the central fraction. So for example, if you can get a little lower recovery, you can cut away some sides of the peak and you will get higher overall activity. Or, for example, also we got size separation. So different peaks have different subfraction have different sizes. So during the elution, the size of the LNPs increased. So if you would like to achieve very, very high tuned separation, you can also consider to add this separation to your purification. Another thing is the lipid analytics. So also this was analyzed on our analytical column, so C4. And we see here these were analyzed different samples. So lipid mix, so crude LNP, so this is what comes out from the encapsulator, still containing ethanol and all the impurities, and then different fraction I was showing before on the chromatogram, so from the start to the end of the chromatogram. And interestingly, we see that this different subpopulation of the LNPs that we were purifying did not change in the lipid composition. So throughout the purification, the lipid composition was stable, also in the crude LNPs, but in the lipid mix with the starting material, it was a bit of difference. So a significant difference in the lipid mix, and then the sample and the fraction had the same lipid composition. What is actually very important, showing you that the whole fraction will have the same lipid composition. So if I move again back to the multi-cargo LNPs, so this is the example I showed before for the activity where we encapsulate pDNA and mRNA at the same time. So we analyze this also on the PATfix Switcher. And here is the chromatogram, so loading, washing, elution. But in this peak, we cannot see what is with the payload. So we collect this fraction and send it to the analytic department. Then they gave us this result. So we got here the chromatogram for the non-encapsulated and encapsulated nucleic acid, so we can actually distinguish between mRNA and pDNA in each sample. So it not only gives us a complete nucleic acid concentration, but actually for each payload that is inside the LNP. And we can also follow the encapsulation efficiencies for each separate nucleic acid. So we see here mRNA and here the pDNA, so there was no difference. Both were good encapsulated. Adducts separate which are coming from the mRNA, which are coming from pDNA, and also the concentration of each of the nucleic acid inside the population of this specific LNP purification. So this is the example how it's very good to integrate also analytics already in the process development, and then later in the manufacturing stage. It will really help to speed up the process and to deliver better products. I will slowly conclude, take-home messages. We are showing alternative to the TFF purification. So we use the monolithic chromatography, and hydrophobic interaction is current choice from our side, our recommendation. We got high recovery and like good uniformity of the sample and activity. This is due to the low shear force process that I was mentioning. We showed how this process is scalable, and also in these slides I showed some screening of the subpopulation, so separation for sure can be also tailored based on your specific drug product on the columns. So the time of the operation, haven't really mentioned that, but you could see already on the previous presentation that chromatography with monoliths is fast. And also the capacity is very high, so you can get high % of the encapsulated RNA in the final sample. It is the most important also to add analytics already in the process development. So if you use the PATfix LNP Switcher, you will be able to get very efficient process development for your drug product. So I would like to thank my colleagues from the process development of viruses and nanoparticles, mainly Anna and Tjasa did the experiments presented here. Process analytics department, Tristan and Anna, that are analyzing our samples on daily basis, so thank you. Process development for RNA and DNA that are producing our great RNA of great purity, and also the cell-based assay, Petra, who is doing the luciferase expression experiments. Thank you for attention. If you have questions, I'm happy to answer.
Thank you, Moica. Who would like to start with a question?
Thanks a lot. On top of the things that you showed with in vitro expression, we have worked a bit in parallel with the same column, and we did purification on animal experiments, also with luciferase. And essentially, we saw no difference between the purification methods. So it seems like there's also no adverse effects for the expression and the safety in mice, at least. So
Great.
nice.
Thank you for sharing these insights.
Yeah. When you're eluting your LNPs, there is a little bit of carryover of the binding buffer, obviously.
Yeah.
That in these cases is citrate, I think, mostly.
Yeah, citrate or other cosmotropic salts, yeah.
Yeah. Is that generally not problematic to have in the final drug product? Or are you thinking to add some kind of step to remove the citrate from the final formulation, or should we just leave it in there?
Yeah, we try to leave it in because we want to avoid another process after the chromatography, and usually there is no problem for the citrate or other cosmotropic salt for the final formulation, but of course it's up to the client that is developing drug product if wants to also change at the end, or maybe it has too high concentration already in the elution and it can already regulate with the diluting the final sample that can reach high concentration and so on. But yeah, we did also some stability studies in the meantime, and we showed that samples in those buffer are stable in different temperatures for quite some time, comparable to TFF. But yeah, of course, the final call is always on the producer. Yeah.
Thank you for this nice presentation. When you showed your separation, when you washed the particles away from the column, you showed that there's a fraction before the main peak and after the main peak, which is slightly lower and higher in size, but the activity was quite different. So the activity was quite a bit lower. How do you explain that? I mean, normally, such a small size should not justify such an activity change, and although it goes in both directions. So why is E1 and E6 so much lower in activity than E4?
Yeah, this is actually what we try to understand. So we got certain separation here. We know it's different activity, different size, but the real mechanism is to be studied. So we already talked before about some of your techniques or so on. Yeah.
Anything like, I don't know, surface properties or pegylation? Oh, yes,
maybe answer this. I think E1 and E6 both have very low RNA concentrations. So we have speculated before that E1 could be emptier particles, let's say, of a different morphology. And possibly also for the E6 fraction, but yeah, it is still a question.
E6 should be fuller than
Well, is it necessary that the bigger it is, the more full it is?
So, okay.
Yeah, but at the same time, like the expression here was regulated by the amount of the RNA. So it is something more than that. But yeah.
Interesting.
Yeah,
I just want to mention that this is not huge difference. So these are fluorescent.
Yeah, so it's, let's say, about 10 times difference.
One log.
Yeah, one log.
But that's the threshold, so everything below, okay, but...
Yeah, I mean, this is what we are saying, like, you can do simple step gradient, collect all, like, no, but if you want to really study subpopulation, there are also different particles that can be separated on the column.
Right. Maybe a quick one.
I can send you samples.
So the upscaling, so you go from, I don't know what, some micrograms to, what was for 150 grams?
Yeah.
How far did you verify that?
Yeah, we verified that on the, let's say, 10th milliliter scale. So 40 liter is yet to be verified. Yeah. It was verified with other particles, so we are confident it will be sure also for the LNPs.
Okay, thank you.
So a little bit on this topic, I just have a question, because if I understand you correctly, you had to compensate with the amount of mRNA, if the mRNA concentration was lower for E6, for instance. So you added more particles, right?
Exactly, yeah.
Did you try to do response curve to measure essentially the potency in the different points of concentration? Because you might be saturating with the particles, you may saturate the entry simply.
Okay. We just checked for the toxicity, for example, and there we didn't see difference in the amount. Like we could transfect higher volume, so more particles, but we didn't see the toxic effect on the cells. It's not really answering your question, but partially maybe.
Yeah.
But could be good continuation to. I mean, we have to study this more in detail, yeah.
Yeah, no, it's a good sign that it wasn't toxic. No. Thank you.
Any more questions for Mojca? No, then I suggest we have now a break, and we will continue with our last presentation from Jacek Lubelski at 3:10. In the meantime...